presence. And in the course of writing up the follow-up paper, Calabrese and the new graduate student actually said that these findings could not be used because, in fact, they were uninterpretable. And they were uninterpretable because the control group was aberrantly low. And unbelievably, in the discussion of that paper, the authors blamed the low reading on the inexperience of the investigator, which must have been, it wasn't Stern, it must have been the new graduate student that was being criticized right there. And so what happened was that then there was a follow-up experiment, and the same problem had another aberrantly low control group. And then there was a third follow-up experiment, and this time there was a problem with actually aberrantly high response, way beyond what a linearity response would have ever shown. And so what happened was that Stern decided to combine all these papers into a follow-up, which he then publishes in the Journal of Science, the most prestigious publication in the world. And he publishes a one-page summary. This one-page summary takes the first two papers, which was an acute study, and then the Calabrese study, and then the three follow-up papers. And what he does is he never shares with the scientific community the fact that, in fact, that he had affirmed the validity of the control group in the Calabrese study. He, in fact, criticizes, goes back to the same criticism. It was really an invalid criticism of the Calabrese control. Then he takes the next two studies in which had aberrantly low control groups, and he claims that they were normal. And he combines all these into one, and he tries to then make the case that, in fact, his overall data support low-dose linearity. He never shares, in fact, with the readership that he had, how he resolved the control group with Calabrese. He never shares with the readership that he had, in effect, found the data in the subsequent papers uninterpretable. They all, within a year, became interpretable. Now, to me, one of the big issues was that all this was summarized in a one-page table in the paper, one-page paper in Science. The co-authors, that is Stern and this woman, Delta Uphoff, the graduate student that he criticized, they stated in the final statements of the Science paper that they would follow up with, in fact, a detailed accounting of all their methods so that all the data would be available for the scientific community to evaluate. As it turns out, Stern never does this. And we are left with a publication that summarizes information, ignores or doesn't show what actually the earlier studies were, and tries to reaffirm the low-dose linearity perspective. What this ended up doing was, in fact, preserving the reputation of Muller, while at the same time allowing low-dose linearity to be promoted, and at the same time then also essentially criticizing the threshold dose response model. These papers then became, the Science paper, just because of its, the prominence of the journal, became widely cited. The initial paper with Warren Spencer and Kurt Stern became widely cited. The paper about Caspary's work that became the controversial paper because it was discredited in the Science article, actually became poorly cited. And so it achieved, all this was achieved the objectives, I think, of Kurt Stern and Muller, in that they were able to keep themselves on message, which was support linearity, criticize threshold, and preserve Muller's reputation. And ultimately, when in fact the decision was made for recommending what the nature of the dose response should be for ionizing radiation in the low dose zone, it was dominated by research with Drosophila, and the best dose response information was actually that which came out of the Kurt Stern laboratories during this Manhattan Project funding period. So in effect, what I call scientific cover-up and the misrepresentation actually metastasized into having a very powerful and strong influence because especially Muller was a member of the first National Academy of Sciences Committee, and that committee basically just reaffirmed the conclusions that came out of the Kurt Stern research that we have just discussed. So it had a profound impact on those recommendations, and those recommendations were crucial. 1956, there was this dose response revolution in which the basic dose response for risk assessment was altered from a threshold to linearity. And now that was just for germ cell mutations, but within a year, other national committees and international committees started to generalize this from germ cells to somatic cells and to the concept of cancer risk assessment. And that's how we have stayed with this perspective from 1956, 1957, right to the present time, over 50 years. 1977, the U.S. Safe Drinking Water Committee of the National Academy of Sciences essentially plucked the same reasoning from the same document from the National Academy of Sciences in 1956 and said, this is how we think chemical carcinogens act. And they adopted a linearity at low dose perspective there, and the US EPA has in fact applied linearity at low dose to dozens and dozens and dozens of chemicals since that 1977 publication. And it all goes back to the Kurt Stern research, it goes back to the Calabrese paper, it goes back to the influence of Muller and the cover-up and the not showing the data, and ultimately influencing the scientific and regulatory communities in this country. And this is really our history and our legacy, and it's one that had been suppressed for over 50 years. And that's my story. One aspect of the story that should also be added in, and that is that if one were to take a look at the Calabrese and Stern paper that was the controversial paper, it's very, very interesting because the entire discussion, a 6-page discussion, was entirely devoted to telling the reader that they should not accept the presented findings of the paper until these findings could be not replicated, but until we could explain why these findings differed from the earlier acute study that was done under the leadership of Kurt Stern and done by colleague Warren Spencer. Now, that study, as I indicated, demonstrated what appeared to be a linear relationship. This study, a threshold relationship. One was an acute exposure, this was a chronic exposure. Now, there were 25 or more major differences between these two papers methodologically. It was difficult to explain how the studies, how you could resolve the differences. The one thing that you did know is that there were significant improvements in the conduct of the study by Calabrese in terms of temperature control, in terms of other aspects of protocol. But also what you had too was differences. For example, the dose rate, it's about 15,000-fold lower dose rate in the chronic study versus the acute study. The diets were fundamentally different. There were, as I said, 25 other or so methodological differences. You couldn't explain them. And what, in fact, Calabrese and Stern were trying to do in their paper was to set up a straw man. That why would you even try to publish your data if you essentially didn't want anybody to take it seriously? They didn't set the same criteria up for the Stern and Spencer study, but just for their own. And why was that? Because they probably were concerned mostly with preventing the issue associated with the challenge to linearity. Now, one other aspect of this, and that is that in most scientists' perspectives, I don't know of anybody really who's submitted a paper to a journal that tells the reader don't take the data seriously. But that's what this did. And so, well, how could you get that published? Well, the only way that you could get something like that published might be in a unique situation. Well, now what could be that unique situation? Well, they sent it to the journal that the author was the editor of. And so Kurt Stern was the editor of Genetics, which was the publisher of that article. And now the other interesting thing was that both of these papers were sent on November 25th of 1947, and they were published in January of 1948. The timing is so close. Given snail mail and other circumstances, I doubt that either paper actually was formally peer-reviewed. And so you have a situation here in which the studies that actually underlie much of our dose response and risk assessment practices have such a clouded past that has not hasn't been understood in the past, that when you do hear it, it comes across as something that's hard to believe, but there's a strong factual trail that ties all these things together, and it undercuts the credibility and foundation of low-dose linearity, both for chemicals and for ionizing radiation. That's our history, and needs to be confronted, and it needs to be dealt with. Thanks very much.